during my postdoc, I started shifting to environmental sciences, agricultural stuff. And today I am at the Hebrew University at the Faculty of Agriculture, Food and Environment. Okay, so let me share my screen. Thumbs up if you can see. Yes, all right. 1 second, I want to see your faces, so yes, all right. All right, so patterns in survival enhancing mechanisms derived from plant water storage. Let's see what this is about. Okay. Okay, so I'll start with the guiding questions of this talk. One of them is, can we find patterns in the water management of all plants? I mean, I'm not interested in this plant specifically or that other. I want something very general. And the second question I would like to ask today is, what is the simplest description that manages to capture plants' most basic behavior regarding water? Okay, so two very broad questions. And plants can act in many, many ways. And here are 3 ways. For example, they can have, let's see, this works. Yes. All right, so they can regulate their stomata, open and close their pores to let water out of their leaves. If the plants are, if there's a drought, the plants can suffer from loss of conductivity in their xylem, in their inside in the, well, I'm assuming maybe, yeah, I hardly see any faces, but if you did that, I would know that, oh yeah, sure, I know what xylem is. So xylem are the pipes inside the plant that bring water from the roots to the leaves. But I'm not talking about not this behavior and not that mechanism. Today, I want to focus on one mechanism only, which is the internal water storage in the plant. Okay, so here I have a plant, a tree, and in the simplest description, I have water coming into the plant, and that I will call sap flow. So sap flow is the flow of water in the xylem. But here, when I measure at the trunk of the tree, I will assume that's the water coming into the tree, and there's water coming out of the leaves, and this we will call transpiration. And imagine that the tree is a black box, 1 arrow coming in, 1 arrow coming out. Now they are not necessarily in balance, those 2 fluxes, the transpiration out and the sap flow in. So their difference I will call recharge. So when the difference is positive, when there's more water coming in than coming out, so water will be accumulating in the tree. And the opposite is true. When more water is coming out than in, then the amount of water stored in the tree will go down. And that's pretty much my very first description of a plant and water coming in and out. Okay, so I'm talking about the internal water storage, the water that's stored in the plant. So here I see, I made this graph from real data, but I show it like a cartoon because I want to show you the qualitative behavior. So here I have on this axis, I have the flux, and here I have time. Okay, so the transpiration is water coming out. So early in the morning, this is 6 a.m. So transpiration, water starts to go out of the plant, and it outpaces the amount of water uptaken by the tree from the soil. Later in the day, around midday, then it flips. Then the amount of water coming out of the leaves, it goes down, and then there's more water coming in than coming out. Okay, and this is from real data. Okay, so those are the 2 basic fluxes. So let's find a few patterns in this cartoon picture here. first of all, we see that those two fluxes, they peak at different times. They don't go together. So I find a time lag between those two signals. The second is they have different relative amplitudes. At least for this one cartoon, at this specific day, the transpiration peaks higher than the peak set flow. There's something interesting going on with transpiration during midday, around 12. So it starts, it goes up, it peaks, and then it goes down. And then later in the afternoon, it goes up again, and then goes down. So this dip here is called midday depression. Excellent. But if the tree were a tube of water, and exactly, so like a pipe full of water, everything that happened at the top of the tube would propagate instantly downwards, and we would see the same behavior everywhere in the tree. Not only the sap flow is delayed, but this dip here, we don't find it. It's quite smooth here. Okay, so that's another thing that we find here. And finally, when there's more water coming in than coming out, then this area here will be recharged. So my black box, my plant is being recharged. The water will be accumulating in the tree. And the opposite here in the orange section here, there's more water coming out than coming in. So we'll call this the depletion of the storage in the plant. Okay, so from this one figure, we find a lot of behaviors, and those are exactly the same, the behaviors I will want to investigate. How can I explain that? How difficult is it to explain those features? Now, some motivation for what I'm going to do. I'm talking about flow of water through a plant. This will be especially important in plants that are suffering from droughts, or in regions that certain part of the year will be drier. And we see that we have predictions that in the future, the many regions around the world will become drier. So regions that today are not too dry, they can become drier. So plants in the natural ecosystem can become drier. And specifically, the cartoon picture that I showed before is from real data, specifically from the Yatir forest in Israel. This forest you see in this airplane picture. We see half of the picture is the forest, which was planted around, I don't know, 60, 70 years ago, 60, something like that. And in the top part of the image, we see the desert. So that's in the middle. If you imagine Israel is it's a long country in the north-south direction. Kind of in the middle, there is this forest at the edge of the desert, and there this forest was planted almost exclusively of Aleppo pines. And those trees, they suffer from extreme drought because we have a Mediterranean climate here in Israel. And where this forest is, it will not rain at all for 8 months a year, something like that, some 7, 8 months, not a drop. So how do these trees cope with the extreme lack of water? So that's for motivation. So a few words about my modeling approach. I think that today's topic would be simple modeling approaches to describe, I don't know, natural phenomena. And this horse is a beautiful horse. We see its color, its details, the shine in its hair. This is the full-blown, in the analogy I want to bring here, the full-blown beauty and complexity of a natural system. What I do is that I look at the horse and then I do this. Okay, now this is my horse. I try to capture the most basic features of this horse, and I go with it. So you might see that, okay, it doesn't even have 4 legs, but I don't care. It has legs. I don't know, I think anyone could recognize that well, this kind of look like a horse, and that's good enough for me. And the rest is details that one person might be inclined to add or not. In my case, not. I like to see this kind of horse. But that's my approach. So if you know a little bit more, and I'm guessing that a lot of you know a lot more about plants than myself. That's a safe assumption. If you feel that I am oversimplifying, then it's fine. You're not crazy, because think that I'm trying to describe this kind of horse. But I still find some value in this description. So where's the value? Okay, let's go to the meat. All right. Okay, so this is my model. I don't have any equations to show, although they exist, but that's enough for us. So I have a tree. I have this symbol psi. This is the water potential, or you can think of pressure of the water in the soil. And this is the water potential in the leaves. And like you see like resistors here, because there is a strong analogy between the flow of water and the flow of electrons in a wire. And this is, everyone does that. So we are describing the flow of water up the tree. And now the water potential is lower in the top than in the bottom, and that causes water to climb the tree. All right. And these resistors here, they are drawn here because water doesn't flow for free. There's some internal friction there. And this is why or in terms of plant behavior, this is the hydraulic conductivity of the xylem. Okay. And somewhere in the middle of the tree, there's an X here. Somewhere I put a water storage unit. Now that's a huge oversimplification, right? There's water all over the tree, stored everywhere. But I want to, for now, to assume there is one compartment, one representative compartment of water somewhere in the tree, here. And here water is stored. So when I talked about before the fluxes in and fluxes out, water accumulates or water is depleted from this black box, this is it. So this is where water accumulates or is depleted, somewhere. And for now, I don't care where. So if you know about plants, oh, is it in the phloem? Is it in the leaves? Is it in the root? I don't know. And right now, I am claiming this is not too important for my goals. Okay, so because I have this model here, I can translate this model into an electric analog. So this is the same picture we saw before. And you see the same resistors we have here, they are here. All right. Now this box here where the water accumulates, now in the electric view, it becomes a capacitor. A capacitor accumulates electric charge. Well, it looks like a box that accumulates water, and that's pretty standard. And my model here assumes that there's some water potential at the leaf and in the soil. And in the electric analog, it becomes a voltage supply that changes with time. So those two figures, they look alike. And I just made the necessary changes from the hydraulic to the electric picture. I go one step further because once I have the electric description, then those are just wires that conduct, well, you can think of water or electrons. It doesn't matter right now. But if they are wires, then it doesn't necessarily have to look like a tree. So I can get this part would be this one. And this is this. And this one becomes this. I just bend the wires because they are wires and I can do that. And I have those groundings here. So they are all here. And this doesn't look like a tree at all. But my claim is that if you were convinced that, well, this kind of looks like the hydraulic description of a tree, and I know the rules of conversion from the hydraulic description to an electric description, so this should represent exactly the same system. And I will be delighted if you want to ask me later about details about how to translate from here to there and the equations and everything. Sadly, I will not be showing this right now. I'm aiming at a broad viewership, but with this model, any of them, they're all equivalent. Then I can write equations, and then I can solve them. They're pretty easy and straightforward equations. There are 4 algebraic equations and 1 differential equation. Pretty easy. Linear differential equation, solvable. Good. All right. So let's see the results. One result. Okay. I assume here that the water potential in the soil will be constant. So you can assume that, I don't know, I'm looking at a short time intervals, not of months or even weeks. I'm looking at, I don't know, hours. So let's say the water content in the soil is kind of fixed in this short time span, and the water potential in the leaf will oscillate like a sine wave. Why a sine wave? Well, think of the diurnal forcing that the tree experiences from the atmosphere. It goes up and down and up and down. So it is like this. But because I'm a modeler and I can play with what I built, I can change. I can make this sine wave really long in time or really short. Like why? Well, you'll see why, but right now, because I can. So, and I want to ask what will happen to the flows of water? What will happen to transpiration? What will happen to water uptake from the soil, the sap flow, and what will happen to recharge? There are 3 flows of water in the. All right, so let's start with transpiration. In the transpiration, you see in different shades here of green that, I don't know, the dark shade gives this and the lightest shade gives like this. They all oscillate. This is the response of the transpiration for different frequencies. And what I want you to notice here that they all have kind of the same amplitude. The top and bottom, they are kind of the same for all of them. And that's enough for me right now. But that's not true for the sap flow. So sap flow is water coming in the tree. So if you have a very long forcing in time, then the amplitude will be large. But if your forcing is pretty fast, like, so you'll see this, and this amplitude is tiny. So it does not respond like transpiration responded before. So we see that when we have high frequencies like those, they barely appear in the sap flow, but the low frequencies do. And the opposite picture is true for recharge. So recharge is water coming in the storage compartment. When it goes, well, when it's in when it's above 0, right? Out when it's below 0. And here the picture is the opposite. When I have a very low frequency, then the amplitude here is tiny. But when I have a very fast oscillations here at the leaf, then it responds really well. All right, so each flow responds differently to different frequencies. And I can do the following. Here in this picture, I have the frequency of the forcing and how much the amplitude of response, if it was large or tiny. The green here is the transpiration goes here. So no matter what's the frequency, the amplitude is high. That's good. So the plant is able to respond to whatever comes. Now you might be asking, why do I care about, I don't know, it's 24 hours. We know what's the length of the day. But I'm thinking of the following. We can add any signal. I can Fourier decompose into its basic frequencies. And so any signal is made of long and short waves. So if I think like that, so it is warranted to think of all sorts of frequencies. Now recharge here, we see that when we have low frequencies, it's really low. So they are filtered out. And when the frequencies are high, then recharge, which is this here, is high. So it is a high pass filter in the language of, I don't know, engineering, maybe. It's a high pass filter because this is a high frequency and the high frequencies pass. They're not filtered out. And the opposite is true for sap flow. There's a low pass filter. So for low frequencies, we see the amplitude is super high like this. And for higher frequencies, they're filtered out. Okay, so let's go back to the cartoon pictures we had before. Here we see the relative amplitudes of transpiration and sap flow. So we see that we found that the peak, the transpiration peaks higher than the sap flow. Well, does this make sense? Yeah. Transpiration here, you see that this curve here, the green curve, is always higher than the blue one, always. And that's the amplitude. So we do expect to find that the relative amplitude, they will be different, and consistently transpiration is expected to peak higher. Not only that, we talked about the midday depression, this feature here that at the middle of the day, you might find a dip in the transpiration, but the signal of the sap flow is much smoother. Why? Because as we see here, the sap flow is it filters out high frequencies. High frequencies do not appear in it. And this means that you need high frequencies to make bumps like this. If you only have very long, smooth frequencies, then you get a smooth signal. In order for you to do something like jagged like this, you do need the higher frequencies, but they do not appear in the sap flow. So as you go, if you imagine, I don't know, you have the tree here, and if you measure the flow here or the flow here, the lower you go, and if you can measure the signal, the diurnal signal, the smoother and smoother it will get. It will, all the crazy things that go about in the leaves, they get smoothed out. Okay, so that's one feature that we can already learn from this model. The model is extremely simple. I didn't mention that. It's not only the model is, it's a small model, it's a minimal model. It is the bare least you can do. If you touch it, you break it. You can put more, but you could not have less. Okay, so another feature we see here when we study this model is that we can find that we have in blue, we have the transpiration, and the transpiration peaks before the sap flow in green. Okay, so here in the top panel, I made a situation where they differ by, I don't know, 3.5 hours. And if I play with my model parameters, I can make this difference become 1 hour. What's the difference between those two? Well, I played with the capacitance. Okay, so I did not discuss capacitance before. Capacitance is the ability of the storage unit to hold its water. It can hold tightly to its water, or it can hold loosely to its water. The higher the capacitance of the water storage, the larger will be the time lags. So if we go back to the cartoon picture, we saw before that transpiration peaks before sap flow, and so this green signal, well, the blue signal lags behind the green signal, and we can measure this time here, right? And this time will be dependent on an internal trait of the plant, how hard the tissues in the plant hold their water. Another feature that I would like to discuss is the following. I said that I have this storage compartment somewhere, it's representative, and it's somewhere in the tree. So I made this X for xylem. And I can measure here the water potential in the xylem, in the middle of the tree. Okay, so what is the tension of the water in the middle of the tree? Now, trees will suffer a lot from low water potentials. If the climate is too dry, they will dry out and they will die. And one process by which it happens is that this water potential is so low that bubbles of air form inside the xylem, and that's called hydraulic failure. So that's very bad for the plants. So let's see what the model has to say about this behavior. Here in this graph, I have the water potential here, psi x. So the water potential in the middle of the tree. as a function of time. And it goes up and down during the day. Now here I have three curves, each for a different value of the capacitance of my storage unit. We see that when we have no capacitance, so the water, effectively there's no water storage in the plant, whatever comes out immediately comes in, then the oscillations, the diurnal oscillations are very large. It goes very high and very low, right? Very low is this, the low part, this is the problem here. When the xylem has very low water potentials, this is when hydraulic failure risks the most. Okay, as I increase the value of the capacitance, we see that it still oscillates up and down, but with a smaller amplitude and specific, so it goes less high, but it less low as well. And specifically, this is less dangerous than this situation, right? So the lower the plant xylem potential dips, the more dangerous it is. So we find that the having a storage unit in the plant helps the plant against hydraulic failure. And last but not least, it's I go back to this cartoon figure here, and we see that we have this transpiration. This is water coming out of the plant. So this is the green curve, and I just painted it in purple, the area here. Now, part of this area can be explained by water coming into the tree. If exactly these two curves were one on top of the other, we could think that every drop of water coming out of the plant is coming, another drop is coming into the plant, and they balance each other exactly. But that's not true, because those two curves, they don't overlap. A portion of the daily transpiration will not be coming directly from the soil, but it will be coming directly from the storage unit. It's like a bank account. So I go into that a little bit, and later in the day, I will fill up my bank account. So now the question is, this is the depletion, right? How much the storage unit was used in order to provide readily accessible water when the tree needs most, right? The tree wants to transpirate a lot during the early morning hours in the Yair forest when it's not too hot, but the soil is dry, so it can't provide, so it will use its own water. So what fraction of the daily transpiration was from the storage unit? So with the help of the model, we can do that. So this is the fraction, I don't know, from 0 to 1, or a percentage if you multiply by 100, as a function of the capacitance. So if there's zero capacitance, if there's no storage unit in the plant, so exactly zero water of the daily amount will come from the storage, but because there is no storage. And as storage increases, then a higher and higher proportion of the daily water will come from the storage. It can be even a significant amount. I put 31% because this agrees with the data we have from the forest. So as much as provided from the storage unit. I might have connection problems. I don't see anyone moving. Is it the case? Yeah, kind of sketchy. But well, anyway, just when my internet is failing, I think this is the time to say thank you for your attention. This was a very broad overview of a few fun things that we can explain with a simple model. And in my mind, you don't need a very detailed model to capture the essence of a system. In the same way that I can think of a horse with a line, a simple line, and it does still capture a lot. And thank you. Yeah, this will be all.
Thank you. Thanks. Can you stop sharing just so we can?
Yes.
Yeah. Very nice. Yeah, I think you can, if people have a few questions, you can turn on your mic or you can write me in the chat quickly.
How long did I take?
I did not count per se. I think it was around 30 minutes, maybe slightly less.
Yeah.
Well, if no one will ask, I will start by just asking. I'm just curious, essentially, what does it mean from what you said? Like, if we look at you know, the effect of different amplitude, different frequencies. What does it mean if now the environment becomes more variable, which would imply, I guess, higher frequencies of these things?
Mm-hmm.
What does that sort of, what would we expect to happen in this story that
so
you showed us?
You
showed this kind of, you know.
Yeah, the scale, the time scale in that graph was, I don't know, hours. Like, so you can think that the daily scale is 24 hours, but you might have suddenly a heat wave coming in. Like suddenly it is very possible that the atmosphere will change suddenly in the matter of a few minutes, right? So this would, and sudden changes, sudden spikes in temperature or humidity, you can only have a spiky signal when you have high frequencies. All right. So when you have the more capacitance that you have in the tree, the better equipped the tree is to withstand anything that you throw at it, right? We saw in one of the curves was that the amplitude of the transpiration was kind of steady. And that's exactly what we would, if I were a plant, I would want that. I would want my transpiration to keep its level no matter what. And that has a price. And the price who pays is the storage unit. So as long as if you have a storage unit capable of doing this, then you're good to go.
And a good storage unit is essentially the amount of water in the plant versus the amount of biomass in the plant or something like that.
Right. So that's a confusing, at first it was very confusing for me. I keep going back in the same words that you said. You can think of the amount of water a tree has in it, and this is super important, but the capacitance is not the amount of water, but how strongly the tissues in the plant hold the water. So if they hold it so strongly that no amount of pressure would make the water come out, then it will be a very low capacitance. So the capacitance is how much energy do I have to put in order to get 1 liter of water out, or whatever quantity, right? So the amount is important, but also the how hard it is to, and it depends where the water is actually stored. If it's in the trunk or in the leaves, they will have different amounts, but different capacitances. They will hold water differently. And I didn't mention, but in my simple model, everything was fixed. The resistors were a number, right? The hydraulic conductance in the tree doesn't change, but it actually does. But if I had that in the model, I couldn't solve it. In the same way, the capacitance, how strongly water is held by the plant, it does change in time. It can change, and it does. And I assume it was fixed. Plants that are drier, they will hold their water more strongly, for example. And this can be relevant in agriculture. I was talking about the Yair forest, a natural ecosystem. But well, the whole idea of irrigating is that the soil stores water and the plant stores water. So if there is a higher, a warmer weather, then the plant that you grow, it can use its water, and then it can, well, you irrigate it, and then it will be replenished afterwards.
Thanks. Anyone has any other quick question? I have more questions, but I think we'll wait for that. Okay, so I think for now, thank you again, Yair, and we'll move along to Lorenzo. So Lorenzo Menichetti, if I'm pronouncing that correctly, is a researcher right here in SLU who did his PhD in soil science here. And I think today he's going to talk more about stuff to do with soil, if I understand correctly. Anyway, Lorenzo, the floor is yours.
Okay, the slideshow. The system I'm going to talk about, it's a lot simpler than what has been taught so far. At least the signal processing that I've seen is very fascinating, but for me it would be an overkill. Mostly I work with systems where the signal is one single pulse. I don't have much oscillations. But I've seen some of the, I think I would share quite easily what Yair told about in the introduction about wanting to find the simplest possible description for a system. And the take-home message of this presentation also for me is that modeling is not going to be about representing reality. We don't have to represent reality. That's not what a model should be. Model, it's you want to predict a certain phenomenon, and this you will see that it doesn't necessarily mean that you want a model that is as realistic as possible. These are the objects I work with. I model soil organic carbon over these time scales, and with this relatively low resolution. I work with annual data mostly. And the system I work with is this one. It's really this simple. And actually, the simplest unit is that you have one single pulse of input, and you model what happens to its decomposition. Then you can put, in annual steps, usually you have, of course, multiple pulses, but each single pulse, it's a signal that you follow. And the conventional theory is probably the simplest possible model. And here I introduced just this small equation just for a bit of history. This linear model was developed already in the 60s. And it's on this approach, all the soil organic carbon decomposition models that you see around, the one that has been used so far at least, they are all based on this this equation. They complicate it a bit, adding more of these carbon pools that decay, but they decay all with still with the same equation. You just compose more of this. And this was developed when there were no computers, and it was working surprisingly well already at that time. This is what is striking for me, that you can describe the composition of soil organic carbon no matter where you are, no matter the conditions, with something very simple. The only complication that you need is a linear scaling of the kinetic to consider temperature and moisture effects. Of course, microbes, they respond a bit. The higher the temperature, the higher the activity, the higher the soil moisture, the higher the activity to some extent. But even this is the equation by Arrhenius, and it's still one of the most accurate. There is a paper from '94, Lloyd and Taylor. They tested how much soil respiration they can explain globally with this relationship. It's something like 90%, 95% of the variance. There are these overarching relationships. And to me, it's fascinating because it seems that soil, this is the assumptions, they hold surprisingly well. The basic assumption is that microbes are only carbon limited in soil. That there is, as soon as there is substrate, they will eat it, they will decompose it with a certain speed. They need to break down the molecules. And this is the only thing that will limit them. So this is limited basically by thermodynamic. The Arrhenius equation is something that was developed for chemistry. But of course, biochemistry is chemistry. this assumption holds very well in experiments. There has been those thousands of experiments in lab nowadays, and also to some extent field studies. This assumption holds because we assume also that soil is microbially redundant. So it means that all the functions are always there. Of course, if one function that you need is missing, the machine is it doesn't work any longer, so it won't follow the same curve. But in most soils, in all soils that I've seen, also this assumption works. And I've come up, I stumbled on this paper recently. They tested on a global data set the diversity of taxonomy of the community, latitude, the Duffy characteristics, and functional diversity. And they found that pretty much everything was following more or less a function of latitude. Functional diversity, no, it was flat. Meaning that functional diversity doesn't change as the other classes change. Of course, you find different guys there, but still they can do the same stuff. Doesn't matter where you are in the world. But then this is the interesting part of my field now. It's like 10, 20 years that there is a lot of debate, which is, it has always been, but now it's like 100 times more intense. And this debate is because we are getting a lot more measurements. That's the first thing that come in. Before we had this lab data and this annual time series. Now we had the flux towers. We have high resolution gas measurements. Spectrometers are more common. You can measure a lot more things, and you are the time scale is getting tighter and tighter. And so the assumptions are starting to show that they don't hold as good as before. And in the theory, we are starting to include a lot of microbial mechanisms, which make the system variable over time. Metabolism is varying, microbial metabolism is varying over time. We are questioning the organic matter protection paradigm. And there is a lot of talk about moving from this linear model that lump just this overarching pattern to something that can do a bit better with oscillations, with the small things that we see. And why should we do it? This is the, if you talk to a microbiologist, it will tell you, he or she will tell you that we should do it because the models that we're using are not representing reality. It's not true that when you go looking into the actual thing, microbes are not just carbon limited, and soil is not microbially redundant. And we find sometimes some cases where this doesn't work. If you put in microbial kinetics, then representing microbes, instead of that just simple equation, we know that microbes are moving the whole thing. So why shouldn't we simulate also microbes? And any experimentalist will tell you that predictions will be more accurate because we will have a more accurate model. This is how we could do it. It's another, the simplest possible system that considers also microbes, which are decomposing, dying, becoming part of soil organic carbon, and decomposing soil organic carbon in turn. They produce exoenzymes, which are the regulation of the decomposition. It's this regulatory gate. Oh shit, it should, the decomposition arrow should grow here, not the inputs. You increase the amount of enzymes, you increase the decomposition. This means that in the equation that I showed you before, this fundamental equation is changed in a very, very heavy way, at least in terms of consequences for models. You have, instead of just one constant decomposition kinetic, you have part of the kinetic that depends on the amount of microbes, so the carbon stored in microbe. So this kinetic becomes now variable over the amount of carbon, which varies over time. It seems a small detail, but the question is more realistic, better. This simple model, it's much more realistic. But the problem is that its predictions are not realistic. We find oscillations that this class of model has oscillations, but they are not present in reality. We've never measured this kind of stuff. There are some attempts to filter them out, adding some negative exponent, adding here and there some other processes. Density dependency is one of the most considered process. But still, the point here is that when you complicate the model, if you miss one small part, one component, and you don't know which component you are leaving out, if you miss one component and that component should have been in, the consequences can make the model pretty unrealistic, like in this case. And this impacts a lot predictions. Because with these nonlinear models, since the consequences of your choices are so big on the predictions, when you use them for extrapolation, extrapolate in the future, the variance of their prediction can vary enormously. When you calibrate them to your time series, and okay, once you calibrate, it's no problem for any model to simulate this very simple decomposition time series. But then when you extrapolate, their behavior can vary enormously. Here you can see in a study, this is the variance of the prediction from conventional theory. And this is what happens if you add nonlinearities in vertical processes or in microbial processes. Here, just summarizing a good way, very simplistic maybe, but for me it's a nice schema of how to classify the different approaches. Autonomous and non-autonomous, it's mostly we work with non-autonomous model. I have a lot of forcing variables. It's not true that I work with just one single pulse. I have temperature and moisture that vary usually. And autonomous model, it's if I have a lab experiment, I can just, okay, temperature and moisture are constant. I have just with this single pulse, but mostly I will have to deal with non-autonomous model, where I have something that just varies externally to the model. And then this nonlinearity, which I talked about. This schema, this is pretty much the whole debate in my field in the last 20 years. And why should we choose one? Why should we choose another? And here I would argue why we should still choose the first, the simplest way, at least in some particular, depending on what we want to do, we should choose the first. I wanted to argue for this. I took this time series, this set of time series. These are sites that, soils that are kept devoid of any input since 50 to 60 years. Some sites are a bit younger, like Askov and Kursk, 20 years only. So here we see really the pulse. It's this organic carbon that was at the start of the experiment. Nothing else has been added. And you see, you can follow the decomposition. There is, it's a simplified system because we don't have substrate interaction, like adding sugars that can boost a bit microbes. In order to test it, I took Q, which is probably the most refined among these linear models. It is a linear model, but it's quite elegant because instead of having these compartments that I showed you before, like you have usually one approach is to have the Young organic method that what comes in from the input, which decomposes relatively fast, that then decays and goes into the older, slightly older, then slightly older and older and older. And you describe this huge variety of molecules and degrees of protection with these discrete pools. Q doesn't have discrete pools. It has one continuous distribution of quality. And instead of simulating the changes in quality by moving the material between the pools, with Q, you are just translating this quality distribution. So it's the most general of this linear model that I know of, because it's neutral towards the assumption that we have 3 compartments, 10 compartments, 2 compartments. Q has all of them, but still linear model. I tested it. This is to explain the plots that you'll see. I work a lot with stochastic, so I explore the parameter space. This is, let's say that my model has only 2 parameters. And instead of finding the only single best optimum, here I would take both. These 2 combination of parameters have pretty much the same fitness. In all my studies, I consider all the space at the same time. And so all my predictions are not one single line. The thickness that you will see in the next plots is explained by the fact that I'm taking all these peaks. These are the parameters of Q. I assume that some will vary within the site, between the site, and some will be general. The general one that I assumed at least are the how quality changes over time and how microbes grow. More or less, I would assume that they are all the same kind of guys, as we said before, the redundancy thing. Then I let some parameter vary by site. This doesn't mean that in the final result they will have to be different. I just let the calibration to be able to vary them if needed. One single site will have, of course, a different initial quality, some edaphic dependencies, and possibly I thought maybe also microbial efficiency. It might be different. And like this, I'm testing, is it really different? The simulations are, this is not really visible. I hope you can see that the model is doing very good. But the time series are a small black line here. Anyway, it's the variance is explained pretty well, particularly these 2 sites, which are a bit weird kinetics. They are explained pretty well by the model, and now we see why. Most of the variation is explained by the initial quality. This means that these sites, for example, Rothamsted and Versailles, we know that they were both grassland before the start of the experiment, so they have a bit of fresh organic matter accumulated, which will decay faster. And this is what you see in this initial quality. This is what normally is called model initialization. And when you talk about organic carbon, there is a lot of talk about how to initialize, which means to get right the initial pool distribution. And just varying these, I would say that we can explain most of it. Kursk was having a little bit, to explain Kursk, we needed a little bit, varying a little bit the efficiency. Kursk, it's a black soil. Processes are slightly different. It's like in the black belt of Russia. So it's accumulating a bit more organic carbon than normal oxic soils. The edaphic parameter was basically not needed. The calibration didn't want. I always attribute a bit of agency to this calibration. I mean, this means that the calibration didn't want to use the edaphic parameter. It was better to concentrate all the information from the variance in the other parameters. And the take home from this is, to me, this means that still we can cut down a lot of details from this decomposition process. And the decay kinetics are pretty much universal. Like I've been fitting a linear model, letting vary only this initial quality, and to some extent a bit of scaling of the from edaphic parameters. And it was doing a pretty good job without the need to complicate more. So the old paradigm, should we abandon it or not? I would say it depends. If our objective is to simulate this kind of time scales, I would say absolutely not. We are just going to look for troubles and without any advantage. this is kind of the final slide. I like a lot to talk about this. This principle, I took it from machine learning, but I think it applies pretty well also to my cases. I mean, you always probably you've been listening about overfitting. This is overfitting. What happens when you overfit and why? The more complex a model can be, the less bias we'll have. Bias is I have to predict my time series, and I see how many points I can visit of my time series. The more complex a model, the more degrees of freedom we'll have. So the more any algorithm will fit it quite easily. You potentially, if you have it complex enough, you will represent all your points. So is that a better model? Absolutely not, because when you go extrapolating with it on a different site, you are kind of bringing in all the biases from the training data set into this extrapolation. And usually it gives a worse performance. I can simplify my model like that. I will get the overarching patterns a bit more, but I will not be able to visit all the points. So I will have higher error, but I will carry on, bring less biases to all my extrapolations. And so there is no right or wrong. I think for every single application, we should try to find this optimum, which will definitely vary. These cures are not really quantitative. It depends on the model. This is quantitative. If you are talking about neural networks, okay, this on the x-axis you have a continuous function, but with parametric models is not continuous. Okay, this was the end, and I was trying to cut it. I think I managed to keep it short.